Okay, let's just dive right into this. Welcome to this explainer, where we're going to treat advanced genomic theory kind of like a biological detective story. And our prime suspect? The goldfish. Seriously, we're going to look at the incredible science behind how one of the most absolutely common pets in the entire world actually represents an unparalleled model of evolutionary and genetic manipulation. So the core mystery we're really trying to solve here is this. How in the world did a completely ordinary, dull gray riverfish transform into the incredibly bizarre, beautiful, and highly diverse creatures that we see in aquariums today? To figure that out, we have to look way past the glass bowl and dive straight into their DNA. When you compare the ancestral wild crucian carp to the modern goldfish, we aren't just talking about, a fresh coat of paint. We're talking about a radical, fundamental restructuring of the animal's entire morphology, going from a flat-eyed gray fish with a basic single tail to an animal with a twin bifurcated tail, vibrant multicolored pigmentation, and these massive bulging celestial eyes. I mean, that's an evolutionary leap that usually takes millions of years. But here, it happened in just a fraction of that time. To understand how, here is our agenda. 1. The ancient genetic accident. 2. Domestication and diversity loss. 3. Decoding those bizarre goldfish traits. 4. The genomics of color. And finally, 5. The CRISPR breeding revolution. Let's get into it. Starting with Section 1. The ancient genetic accident. There's this massive 14 million year gap between a critical genetic event and actual human intervention. Think about that timeline. 14 million years ago, the ancestor of the goldfish underwent this massive whole genome duplication event. Then, fast forward to just about 1,000 years ago, during the Song Dynasty in China, and breeders finally started exploiting this genetic anomaly. And today? Well, today we finally have the chromosome-level modern genome sequencing to understand exactly what those ancient breeders were actually working with. The whole secret lies in a phenomenon called allotetraploidy. No, the best way to think of allotetraploidy is like a computer backup drive. Because of that ancient whole genome duplication, the goldfish possesses two entirely different sets of parental chromosomes. So because one set of genes keeps the fish alive and handles all those essential physiological functions, the other set is essentially free to just mutate wildly without killing the animal. It literally provided the ultimate biological blank canvas. And because of that backup drive, the goldfish boasts an absolutely astounding 70,324 coding genes. To put that into perspective for a second, that is roughly double what you'd find in a closely related diploid fish model, like the zebrafish. This immense genetic redundancy is the absolute prerequisite for the crazy genetic flexibility required for such extreme ornamental selection. But here's the kicker. This genetic redundancy wasn't just sitting there static. It started mutating at a highly accelerated rate. In fact, the data shows that 30% of these retained duplicated genes have rapidly diverged in their expression across different tissues. Plus, they're mutating 1.7 times faster than what we see in salmon, which, by the way, had their own genome duplication event 80 million years ago. This incredibly rapid divergence provided the raw, malleable clay for human domestication. Which brings us right to Section 2, domestication and diversity loss. Now, you might normally expect intensive inbreeding to lower a species' genetic diversity, right? But surprisingly, overall genetic diversity actually increased from the wild crucian carp to the highly modified wen and egg breeds. Why? Because human breeders actively protected rare, bizarre mutations that would have absolutely been lethal in the wild. We artificially preserved the genetic outliers, essentially hoarding all this weird, wonderful DNA. However, when we zoom in on specific regions of the genome, what you might call the genetic scars of domestication, we see massive drops in diversity. These are called selective sweeps. So while the whole genome got more diverse overall, the very specific areas controlling the traits humans wanted, their diversity plummeted, dropping by up to 4-fold. And the genes caught in those sweeps, they perfectly mirror what humans have been hunting for over the last millennium. When researchers analyzed these specific sweep regions, they found them heavily enriched for genes controlling things like behavior and docility, massive eye size, and brain development. Basically, breeders were actively locking in the DNA for tamer fish with strange heads and giant eyes. All right, moving to Section 3, decoding those bizarre goldfish traits. Let's see how this works mechanically. Take the iconic twin tail, which you see in varieties like the Aranda, It isn't magic, it's actually a highly specific breakdown in embryonic development. Normally, a fish's top-to-bottom blueprint is guided by this balanced gradient of bone morphogenetic proteins, or BMPs. But in twin-tail goldfish, a mutation in a gene called chordin disrupts this entire gradient, literally splitting the tail skeleton right down the middle as it grows. But even more fascinating is the biological whodunit behind the upward-looking eyes of the celestial goldfish. Genomic sequencing finally revealed the culprit. It's a mutation in the LRP2 gene, a single premature stop codon, which is essentially a genetic typo that just drops a period right in the middle of a DNA sentence, truncates the protein, fundamentally altering the entire architecture of the eye. And that single mutation triggers a massive cascade of cellular errors. The truncated LRP2 protein leads to severe corneal dysfunction, it alters extracellular matrix secretion, and it kicks off increased inflammatory reactions. All of this absolute physiological chaos culminates in the eyeball swelling and rotating up to 90 degrees skyward. It is just an incredible example of how one tiny genetic typo can drastically change a living animal's shape. Let's shift gears a bit. Section 4, the genomics of color. While mammals basically only have melanin to work with to color our skin and hair, goldfish utilize up to 6 distinct pigment cells, called chromatophores, to paint their scales. They've got melanophores for blacks and browns, xanthophores for yellows, erythrophores for those bold reds, iridophores for reflective shine, leucophores for crisp whites, and even rare cenophores for blue. It is a wildly complex biological palette. But this color isn't just random. It's a highly regulated chemical factory. Transcriptome analysis reveals that this vibrant array is strictly controlled by core metabolic pathways, melanogenesis, tyrosine metabolism, and complex signaling cascades like Wnt and MAPK. By tweaking these exact genetic control boards, breeders can effectively dial up or dial down specific colors. It's almost like adjusting the color settings on your TV. And that brings us to the modern era. Section 5, the CRISPR breeding revolution. Today, the slow guessing game process of visual selection is completely being enhanced by genomic selection. Breeders can extract DNA, run it through high-density panels to find genetic fingerprints, and use marker-assisted selection to target specific traits. By calculating a genomic estimated breeding value, breeders can predict the true genetic worth of a fish based purely on its DNA. They know exactly what traits it will pass on before it even fully matures. But the real, absolute game changer here is CRISPR-Cas9. Scientists no longer have to wait around for nature to provide a lucky mutation. They can directly edit these pathways. By knocking out the OCA2 or TYR genes, they can engineer specific albino or color patterns. By mutating chordin or LRP2, they can instantly recreate those complex ornamental shapes like the twin tail or the celestial eye on demand. They can even disrupt myostatin genes to rapidly increase skeletal muscle mass. We are literally right now typing the desired color, shape, or size directly into the genome. So the absolutely crucial takeaway from all our sources is this: the goldfish is so much more than an ornamental pet just swimming around in a bowl. Thanks to its unique genetic history and that ancient duplicated genome, it is a living laboratory. It bridges evolutionary developmental biology with advanced commercial aquaculture in a way that literally no other vertebrate can. I'm going to leave you with this final thought to ponder. For 1,000 years, we blindly sculpted the goldfish using natural mutations, just feeling our way in the dark. Today, with CRISPR and modern genomic mapping, we have the precise blueprint and the editing tools to match. If we can rewrite the genetic code of a simple river carp this drastically to create a living masterpiece, what else are we on the verge of reshaping in the natural world? Thanks so much for joining me on this explainer. Keep asking those big questions and keep exploring.